Our next speaker is Dr. Julia Laskin. She's the William F. and Patty J. Miller Professor of Analytical Chemistry at Purdue University. Previously, she was a chief scientist and fellow at the Pacific Northwest National Laboratory. And her research into bioanalytical chemistry, spectrometry imaging, and technology development covers wide-ranging applications, including environmental analysis, biological systems, and material science. So I think we're going to hear some really exciting advances from her today, and let's give her a warm welcome.
Well, thank you very much for this nice introduction and for the invitation to speak here. So I'm probably one of the few people here in the audience who does not do single cell analysis, not deliberately, right? So, sorry, yeah. But hopefully what I'll tell you today, so this is still in the,
I know, I'm
yeah.
trying to.
So, but the techniques that I'll be telling you today about, we actually do look at single cells on a daily basis, and we look at them in the environment of a tissue. So the technique that I'll be talking about today is mass spectrometry imaging. So, all right, so let me, then you won't see the wonderful faces of my students that will all appear right there in the corner, but I'll point to their contributions. So, okay, so I'll be talking about ambient mass spectrometry imaging techniques today, and I'll tell you some of the developments that bring them to the spatial resolution that is comparable to the size of the cell, or even better than the size of the cell, which allows you now to study cells within the tissue microenvironment and explore their molecular signatures. All right, so let me talk through some of that. So the idea is to be able to measure the spatial localization of different biomolecules in biological tissues, including metabolites, lipids, and proteins. And you've heard a little bit about those different aspects in the single cell context yesterday and today. So I'll show you what, when we kind of abstract ourselves from the goal of doing single cell analysis, and we still we still do it in imaging experiments. Okay, so so here is the idea. We are combining spatially resolved lipidomics, metabolomics, and proteomics to get those comprehensive molecular maps of biological systems. And the advantage of mass spectrometry imaging is that you can do it in a label-free manner. So you have an untargeted, an opportunity for completely untargeted analysis, or you can make it targeted if you have targets. It's selective and sensitive, and it provides you insights into the cellular communication within the environment of your, say, biological sample or biological tissue. So if you want to do these experiments with the single cell resolution or subcellular resolution, a lot of different technologies are getting there. Some of the most advanced ones are imaging mass cytometry, or the beam kind of ion beam kind of analogs of this technique, where you are bombarding your samples with a laser or with with ion beam, and you have antibodies labeled with lanthanide, and then you measure just lanthanide with your mass spectrometry, so pretty much aerosolize everything out from your sample. And you can do it these days with really, really high spatial resolution. It is a targeted technique, so you have to have your targets. You can image proteins with subcellular resolution, getting very, very beautiful images. From the standpoint of the techniques that I am more interested in, those are molecular imaging techniques where you don't have specific targets, you don't have any labels. And so currently, MALDI and MALDI-2, which is MALDI assisted by an additional laser to enhanced ionization, they have reached the resolution down to about 1 micron in some cases. So this is still a very advanced experiment. It's not a routine measurement, but you can get there. And there are also some efforts that are focused on tissue expansion-based mass spectrometry imaging. So if you can't get a better spatial resolution with your system that you have, let's just stretch the tissue and get the spatial resolution using this approach. The current challenges in this field and where the focus is still on improving sensitivity, throughput to enhance the spatial resolution. There is still a lot of complexity in biological systems. So, and when you don't have any separation, you see all the molecules at once. So we're still focusing on improving the selectivity of the techniques. Quantification is one of the big challenges, and most of the techniques that are available, commercially available these days, they require specific sample pre-treatment, matrix application or such, which may introduce some artifacts which particularly become important when it comes to imaging at a cellular or subcellular resolution. So the technique that we're developing is an ambient ionization technique that you've heard a little bit about. So we'll call it nanospray desorption electrospray ionization or nanodesi. And so it's basically summarized here in a very simple cartoon. So you have 2 glass capillaries or channels in your microfluidic probe, and you run your solvent through one of the channels and you pick it up into another channel and you spray, you generate electrospray here at the end of this capillary. So here is your syringe pump propelling liquid through those 2 capillaries. You make a little liquid bridge here between those 2 capillaries. You extract your analytes into this liquid bridge. You transfer them to a mass spectrometer inlet and analyze by electrospray ionization. And so now all you have to do is to move your sample under this probe. We'll call it nanodesi probe. So this will be our probe. We move the sample around. We acquire mass spectra from different locations. And then we take all the signals we see in our mass spectra, we generate those 2-dimensional maps, and we get those palettes of ion images. So this here in particular, this is mouse brain tissue section, a really old experiment, but just to show that we get hundreds of images in the same experiment. So each of those like little rectangles here is an image of an individual molecule. And what I will be showing you here is the intensity will go from dark to light. So low intensity will be dark and high intensity will be bright. So a very simple concept, by providing this path to the analytes from the sample to a mass spectrometer, we get a very sensitive detection, and that allowed us to reach the spatial resolution down to 10 microns. We've also published something like 5 to 6 microns as well. It's a little bit tricky to determine the spatial resolution in those experiments. We play a lot with the solvent composition, so we would do that for quantification and online derivatization. And we put a lot of effort into the identification using on-tissue tandem mass spectrometry. We currently have this technique coupled to 6 commercial instruments in our lab. We did publish a tutorial on how to modify a commercial instrument to be able to implement those platforms on commercial systems. And of course, there are platforms in the Kelleher lab and other labs worldwide that use this approach. All right, so when we do these experiments with high spatial resolution, we pull the capillaries. So here is my capillaries that I showed you previously, right? So the 2 capillaries that run the solvent, we pull them finally, and they will look like that in reality. So this is our 2 capillaries running the solvent, 1 and 2. And we add a 3rd capillary in between here going vertically. So that's a capillary that senses the distance to the surface. So this is a microprobe technique. So you need to control the distance between your probe and the sample in order for you not to run into some, valleys and hills on your sample and either break the probe or lose contact with the sample. So And so using this approach, and we also have microfluidic probes that do this type of sampling, which I'm not going to talk about today, but we can do those imaging, we can perform those imaging experiments either with this type of capillary probe or with a microfluidic probe, and we can get a spatial resolution down to 10 microns. And this is just to show you some, to give you some idea of how the data look like when you are imaging with high spatial resolution. So this is a small mouse uterine section. It's just a little over 1 millimeter across. It has distinct cell types here. So the dark kind of branch type of structure is one type of epithelial cell, so luminal epithelium. You have those kind of dots around them. Those are glandular epithelial cells, and you have stroma surrounding those. And when you look at images of phospholipids here, shown here, some fatty acids and phospholipids, and I give you the kind of key to read those annotations. So we look at phosphatidylcholine, for example, so PC, with 36 carbons in combined acyl tails and 4 double bonds. So this is our nomenclature. And you can see very different localizations of those species, right? I wanted to point your attention to those 3 sphingomyelins. We usually see very few sphingomyelins. But those 3, one is localized to luminal epithelial cells, one is enhanced in glandular epithelial cells, and one is present in both types of epithelial cells. So you can see it all the time. Lipids of the same class, just sometimes differing by the number of double bonds, show you very, very different localization. You can also see something that you would usually attribute to just insufficient spatial resolution of your experiments, but in this case it's real. So those fatty acids, for example, they're not very tightly localized to the epithelial cells. So they're spread out, right? And so we see those kind of shallow gradients in biological systems a lot, which makes it kind of difficult to define exactly the single cell location based just on mass spectrometry imaging data, because biology does not like steep gradients. There are usually no steep gradients in biological systems. All right, so you heard a little bit already from Jared about imaging of proteoforms, and I just wanted to show this early work that was done in collaboration with the Kelleher group, where we were able to take a tissue section, remove lipids from it, and then image it for proteins. We did that with kind of regular probes, not very high spatial resolution probes. We also showed that this works with high spatial resolution probes. What we're able to lean on is that the technique, this nanodesi at the end, ionization, is very similar to electrospray ionization. So you generate multiply charged ions, and then you can use your top-down proteomics approaches to identify them. So we do on-tissue MSMS, and we identify all of those molecules in imaging experiments. So we have those capabilities, and I just wanted to show you some examples from this very early work that we did, the first demonstration that this experiment works. So this was mouse brain tissue, and we washed it, we analyzed proteins in this tissue. We also did top-down proteomics and identified multiple proteins. We got those different maps of different proteins in mouse brain tissue. Of course, those are quite abundant, so those are some of the most abundant proteins. But nevertheless, we were able to observe 40 different proteoforms, which you cannot do using optical approaches. And so 40 different proteoforms of 19 proteins in this rat brain tissue. And then we also extended this approach to higher spatial resolution, which is still quite challenging, but we can get down to 10 micron resolution in these experiments. So I just wanted to show you what you can learn about different proteoforms by mapping them spatially on tissues. So those are proteoforms of myelin basic protein in brain. So you see, I'm just showing you a subset. There was, I think, 8 or 9 of those that we saw in one experiment. So you can see the localization of those proteoforms in brain. Here are their annotations. And it didn't look very exciting when we saw those data, but then we plotted ratio image or fractional images of those proteoforms. So what we do is we take the signal of this proteoform and divide it by the sum of the signals of all of the proteoforms of myelin basic protein. And when we do that, we see some really interesting things here and some indication of some cellular kind of, of enhancement of those specific proteoforms in cells that are present in a specific environment. So here is an example of an acetylated proteoform that is very abundant here in the cerebellum region, and it's present also in high abundance in midbrain than some other species. Its analog that also has oxidized methionine is mostly predominantly abundant in the midbrain region, and it's actually repaired by the known sulfoxide present in the cerebellum region. So that's probably why you don't have this proteoform in high abundance in the cerebellum region. You have phosphorylated proteoform that doesn't show any specific preference to any specific cell types. And similarly, this acetylated and phosphorylated proteoforms shows a similar flat distribution in this fractional image. And we'll have some unassigned proteoforms. And I'm only showing you to say that, okay, there are some really interesting localizations in some species that we still don't know what they are. But we really hope that as databases are getting more advanced, we'll actually learn and we'll know what it is. But there are some species that we're discovering that are not in databases. All right, so with that, I told you that we can image lipids and metabolites. I showed you that we can image proteins. So it was natural to couple those two experiments and image both, do this multi-omics kind of spatially resolved analysis of tissues in the same experiment. And so the idea here is that you will section the tissue, and the example here I'll be using will be pancreatic tissues that show really beautiful immunofluorescence images if you stain for insulin and glucagon, and that's what we do. So we acquire an optical image of the section, we stain for insulin, and you see those beautiful pancreatic islets here in this section. We stain for glucagon, and we see this very typical for mouse pancreas, the localization of alpha cells that are surrounding beta cells in the islets. And we take a serial section and we image it for lipids and metabolites. Then we can wash the tissue and re-image it for proteins. And we also take the tissue section that was imaged for lipids, stain it for insulin. We can, in principle, take the stained section and re-image proteins, but does not work that well. But we've done that experiment as well. All right, so of course we were, and then we generate, in addition to immunofluorescence images, we generate ion images that we can then correlate to the cells that we mapped using immunofluorescence. So a big question was, do we actually perturb the system after we image the tissue section for lipids and metabolites? So what we did here, we take and stain the tissue after we imaged it for lipids, and we obtained this distribution of insulin in this section. And then we took a serial section that was fresh, that wasn't imaged, and we also stained for insulin. And you can see that there is a pretty good correspondence between those two images. We looked at the overlay. They're a little bit shifted, just as we would expect in serial sections, but it doesn't look like we are delocalizing anything, which means that we can actually do this type of correlative analysis. So then we imaged for lipids, we imaged for proteins, and we got those very detailed maps. So here, this is not a high resolution imaging data, but this is the only one where we imaged for both lipids and proteins. That's what I'm showing you, that I'll show you high resolution images later. So we stopped imaging for proteins just because it wasn't very interesting in this system, because you only see insulin and glucagon predominantly. There's nothing really particularly interesting. You do see also hemoglobin, but again, it's just marking the blood vessel. So it's not very exciting. But nevertheless, so this is what you see here. So this is your optical image of the section. You see 1, 2, 3, 4, 5 islets, pancreatic islets. You see them stained for insulin and glucagon. So you observe those islets in immunofluorescence imaging. And then you start looking at some lipids and metabolites. So we see some of the phosphatidylcholines that are often localized quite tightly to the islets. We see glutathione, so a metabolite. We see some other lipids. We see some things that we still have not identified, likely peptides that are localized to the blood vessel region. And we know that because they correlate with the localization of hemoglobin. So this is our insulin image. This is our glucagon image. And again, we have some other images that look interesting, show some different localizations, interesting localizations, but I'll tell you a little more about the identification. There are big challenges in identifying things in those experiments when you don't have a specific target. All right, so let me show you. So this is just a zoomed in optical image of a section that was imaged with high spatial resolution just for lipids and metabolites. And when you look at the mass spectra of those pancreatic islets, you see very abundant peaks of metabolites. You see lysolipids here, lysophospholipids. You see signals of lipids. So that's all pretty standard. There's nothing surprising here. We look at the images of those lipids, and we see them pretty well tightly localized to the islets. We know that from this immunofluorescence staining of this section. So here are the islets, and you can overlay those kind of outlines onto those ion images that we see. So we see some species that are just very tightly localized to the islets, and some species that show you the inverse localization. So they are in the exocrine tissue. So either in endocrine or exocrine tissues. All right, so that was nothing really surprising here. We see it all the time. There's nothing special about this. But what was really special in this experiment, that in negative mode, in negative ionization mode, we also, when we dwelled into the data, we observed a lot of peaks labeled in red here, that then we identified that those were deprotonated peptides. All right, so we looked at the localizations of those deprotonated peptides, and we found some really interesting ion images. So again, this is to guide the eye where the islets are. And you can see that some of those species, and here I'm only showing you the mass to charge ratios of those species. I'll show you some identifications of what we could identify here in just a moment. But you can see that some of the species localized again tightly to the islets, just as we saw for lipids and metabolites. But some show very, very diffuse localization, so very shallow gradient that extends from endocrine tissue into exocrine cells. And some show really also some interesting kind of, very few of them do this, but you can see this kind of localization that correlates with the glucagon localization. All right, so a lot of those species that we saw, deprotonated peptides, were identified as C-peptides. And so let me show you a little bit more about that. Again, and I'm not a biologist, so I do my best. So C-peptides are produced supposedly as a byproduct of insulin synthesis, right? So you have your pro-insulin chain, you cleave You cleave it here, you get your insulin released, and then the C chains they were considered just byproducts, and they were considered not very interesting systems until later, maybe about a decade ago or so, C-peptides have been recognized as important systems, and in fact, they do play a significant role in type 1 diabetes, so there is a lot of interest to them. And so, there are two C-peptides that are usually generated depending on where the chain is cleaved. So there's C-peptide 1 and C-peptide 2. And you can also generate some truncated peptides. So some example is if you cleaved here between the 24th and 25th residue of C-peptide 1, you will generate this DES 2529 C-peptide 1, and we see those species in our data. And so we did on-tissue MSMS and a lot of manual analysis. So if anybody knows how to better identify peptides in negative ionization mode, I think I asked everybody I knew would have some experience with that, but we got really not a lot of help with that. So there is really a big challenge there. So we see a lot of deprotonated peptides in our experiments. We don't know what they are. They're very difficult to identify. But in any case, after a lot of manual labor and analysis of tandem mass spectrometry data, we identified several of those peptides, and I wanted to show you their localizations. So here is again, the optical and immunofluorescence images of those tissues, and here are ion images, and just to show you how close or how far away those gradients extend away from the islet, I'll show you the outlines of the islets here. So here are the outlines of the islets. Okay, so you see that a majority of those peptides are not tightly localized to the islets. They are expressed in the islets, and then they're spread out to the acinar cells and exocrine tissues. And so in general, it's known that T cells are recognizing C-peptides, and usually just like the original C-peptide 1 and C-peptide 2. And so it's not quite clear what truncated C-peptides are doing there, and maybe they're distracting T cells from killing beta cells, because there's a very fine balance there between the healthy pancreatic islets and islets in type 1 diabetes. So we don't know exactly, again, I'm not a biologist, I don't know exactly what it means biologically, but I think this is a discovery that you can make without having a target, without having any idea of what to expect from those systems. You just come across that in your data, and then that hopefully can help you generate some hypothesis how those cells communicate, right? And how those different species are involved in this crosstalk between different cell types in tissues. All right, so I still have a few minutes left. So let me tell you a little bit more about how do we enhance the selectivity of mass spectrometry imaging. And so here I'm only showing you examples of our work with lipids, but I'm also thinking that there might be something there that we could extend similar, kind of apply similar strategies to enhancing the selectivity of protein imaging. So here is the idea. So we have lipids that differ in just the location of the double bond. And just by mass analysis, we cannot distinguish between them. So how do we differentiate between those different lipids? When we when we scan the tissue for just mass to charge ratio, all those isomers that differ by the location of the double bond will be all lumped together. So we'll not know how they are distributed in tissues. So the way we approach this is by adding a photosensitizer to the solvent. So as I said, we can add pretty much anything to the solvent, right? So we add a photosensitizer to the solvent, and we run the solvent with this, in this case, the first example, pink photosensitizer, through our probe. We pick up molecules from the sample, and we then irradiate this volume with the laser pointer. And so when we do that, we generate singlet oxygen. So it's photosensitized generation of singlet oxygen that then attacks double bonds, generates hydroperoxides. And hydroperoxides give very nice fragmentation patterns that are very easy to measure using even very, like unit mass resolution instrument. And so now we can do imaging experiment in tandem mass spectrometry mode and use those diagnostic fragments to map the different isomers of lipids in tissues. And so let me show you one example where this was done with high spatial resolution. So this is this lipid hydroperoxide that we generate. So this is phosphatidylcholine with the 16-0 chain in SN1 position, 18-1 chain in SN2 position. When we react this with singlet oxygen, we generate hydroperoxide at mass to charge 814. We do collision-induced dissociation on this species, and we get diagnostic fragments of two isomers of this lipid, one with a double bond in the location at carbon 9, and another one with a double bond in the location at carbon 11. And so now we can just map those fragments as we go across the tissue, and we can generate high-resolution images of those different lipid isomers. So this is, again, mouse brain tissue. This is a fractional image of delta 11 isomer, and it's kind of inverse delta 9 isomer. So you can see this really interesting differences in the localization of two lipids that only differ by the number of by the location of the double bond. All right, so we further developed a universal photosensitizer. So instead of using the one that I showed you in previous slide, can be only used for positive mode imaging. It generates huge interferences in negative ionization mode. So we found this derivative of a porphyrin. So with the carboxylated porphyrin, that is an excellent photosensitizer that can be used for this to implement this approach in both positive and negative ionization mode. And to our great excitement, we were able to image isomeric fatty acids. So this is fatty acid 18-1, and we can profile its delta 9 and delta 11 isomer based on those fragment ions that they generate. And this is just their abundances here in parentheses. We can also perform experiment where we're imaging lipids in both positive and negative ionization mode. And in this case, we had different classes of phospholipids that we were able to image in the same experiment. So we can now look at this isomeric differentiation of those different phospholipids. And some of the isomers are co-localized and some of them are not co-localized. There are different abundances of different isomers. So this just gives you a lot of additional information and insights into the role of those isomeric lipids in biological systems. All right, so I think I need to wrap up, right? Or do I have 2 minutes? Okay. All right. So just very last kind of bit of, I think, the development that I wanted to show you, and that is the implementation of this platform of nanodies imaging on a triple quadrupole mass spectrometer. So a lot of times you do need a high-resolution mass spectrometer to answer your questions. But there are a lot of things that we can do on a much, much more accessible platforms, and we can lean on the sensitivity, selectivity, and speed of those systems to perform targeted and kind of semi-untargeted or semi-targeted experiments. So the idea here is that we will generate a list of what is called multiple reaction monitoring MRM transitions, where you select your precursor lipid and you select its characteristic fragment. And that's all. So you will be having those MRM transitions that you will be monitoring in this triple quadrupole experiment. Then you map those diagnostic MRM transitions, again, across the tissue section. We had to develop codes for visualization of those data. So you basically take and you have to spread each of the, have to assign each of the MRM transitions to its pixel. And then you have your pixel grid, and you can then project each of those transitions onto your pixel grid to generate ion images, just as I showed you before. Okay, so here is an example where it becomes really handy. So these are two lipid isobars that are separated by 0.2 millidalton. Okay, so this, to separate those by mass alone, it will require about 4 million resolution. So that's really out of reach for most of the instruments. But luckily, those two isomers give two fragments that can be easily separated using unit mass resolution of a triple quadruple mass spectrometer. And so by doing so, we now can take those isobaric lipids and map them onto the tissue using their MRM transitions. And we can see there is not a huge difference in localization here, but hopefully you can see that this species, the phosphatidylcholine, is less abundant in the midbrain region of the tissue. So otherwise it would be very difficult to separate those species, but you can do it using a very simple platform. And you can also separate some isomers if you're lucky, if they have distinct fragments. So this is one example of the oxidized fatty acids where you can map them, those two isomers of an oxidized fatty acid using the same approach onto the tissue section. All right, so let me wrap up and say that we've made a lot of progress in this field. There is still a lot to be done. And I think that if we were able to achieve routinely 1 micron resolution, that would be a game changer for this field. And definitely will bring it much more into the realm of single cell or subcellular kind of characterization of biological systems. Some of the key challenges remain in molecular coverage, identification, and quantification. I showed you that hopefully the future of correlative and multimodal imaging is something that a lot of groups have been doing lately, and you will see more and more of that in this field where you can get more additional information from other modalities. That I wanted to thank my group members. A lot of great work. A lot of work that I showed today was done by Manxi Yang and our collaborators who provided us with some interesting samples, the collaboration with the Northwestern group, Agilent and Bruker teams, our funding agencies, and thank you for your attention. I'll be happy to answer questions.
Thank you. Two naive questions. How long does it take to scan a brain tissue slides like you showed? Is it a fast process? I understand there are challenges in data analysis, but to acquire data.
Yeah, so it's not a very fast process, of course. It depends on the spatial resolution and on your platform that you have. So I think usually those experiments run for like 2 to 5 hours roughly. I've seen some people running them for 24 hours. We're trying to avoid that. Yeah, I mean, if you talk to biologists, they want to image, a carpet with a micron resolution. And that's of course, usually out of reach for us. So we cannot image like a huge section in some reasonable amount of time with very high spatial resolution. But we've done pretty large human tissue sections, like human kidney sections with high resolution. That was probably about a 12-hour experiment when you do it, a large tissue with high spatial resolution.
Okay. And the second question, when you detected polypeptides in negative mode, so you were not able to find the same peptides in positive mode. Is it correct?
No, those peptides do not ionize in positive mode.
Okay. Okay.
Yeah, we see other peptides in positive mode.
Even if you synthesize the same peptide,
those peptides do not ionize. No, we had standards of C-peptide, just regular C-peptide. It does not ionize. It has a lot of glutamic acid residues. Okay. It does not ionize in positive mode.
Thank you. Interesting.
Julia, very nice talk. I have a general question about the tissue, the lipidomics for diagnosis. A lot of people using different kind of methodology to claim that they are able to use the lipid to do cancer diagnosis. And what do you think about this? Is there any specificity on the lipid species?
Oh, for cancer diagnosis, absolutely. Cancer is relatively easy to detect using lipid profiles, because a lot of things change in cancer. So yes, absolutely.
Again, one more question. And when you do the different tissue section across these axes, do you see the different distribution of the lipid, or they are quite homogeneous?
That depends on the tissue, right? So in brain tissue, we struggle a lot to find comparable tissue sections because things change so quickly in brain. But if you were to, like we were imaging, for example, muscle fibers, so you have like this long fiber, you can slice it all you want, right? And not a lot of things will change between sequential sections. With the pancreatic tissues, the size of the islet is about 50 microns. And so, again, you may be slicing, like you may lose your islet in the next section. You may not even see it, right? If you were at the edge of the islet in one of your sections, you may not see that islet in the like adjacent or maybe next to adjacent section. So yeah, it really depends on the system a lot.
I actually have a quick question. I think it's really cool that you can get the kind of resolution for the gradients across these tissue slices. Specifically, when you do the tissue expansion form of this method, do you sometimes see like intracellular gradients? I'm not sure if the resolution is that good, but is that a possibility?
I think it is a possibility. So, and I mean, tissue expansion is a relatively new technique with mass spectrometry imaging, so there's still a lot of validation that needs to be done there. But yeah, once you can see with that resolution, you should be seeing the details of this inside the cell. And there was one experiment where I thought that we saw the cell wall, but you know, it was like once. And we never saw that again. So it's a tough experiment to like really get very to get the signatures that just localize to one specific location, either within the cell or between the cells. Nature likes shallow gradients, you know, so there are no step functions in biological systems usually, right?
Gotcha. And I actually have one more quick question. It's kind of broadly speaking. You discussed that one of the main aims is to develop more and more effective bioinformatic tools for identification. Um, do you have a particular omics that you're most interested in having those tools developed for? Metabolites, lipids, or proteoforms?
So we're pretty good at identifying lipids. I think overall it's not bad. Well, we usually the issue, of course, with isomeric lipids, we may not be able to identify which isomer exactly that is. SN1 is in zero position, double bond locations, all that stuff is kind of challenging, right? But we will at least tell you what lipid that most likely is. With metabolites, well, a lot of times it's very hard. With proteoforms, if you have a database, then you can tell what it is. If you don't have a database, you're doomed, right? And so, and the same with those like C-peptides, for example, negative ionization mode, it's not a lot of tools there for protein identification and peptide identification in negative mode. And so then you really, that becomes really a huge challenge. And we see, like in imaging experiments, a lot more species than we report. And a lot of times we do not report those other species because we have no idea what they are. We see a lot of peptides, we don't know what they are. So that's where the challenge is. There's a lot of information in those data that is not utilized at the moment. They're there.
Thank you.
Really interesting talk. I just had maybe a technical question. In some of the ion images which you showed, to me, and I'm no expert in this, it seems like there are vertical striation patterns. What are those?
Are you saying that there is like a gradient going vertically up? Oh,
Yeah.
just lines,
Yeah.
streaks?
Yeah.
So the streaks are usually, if we see the streaks, we usually don't see vertical, we see horizontal streaks, right? So if you see a horizontal streak like that, that means we lost contact with the tissue. So that's pretty standard. 14? Okay, let's see. It's on 14. So I think this is 14. Oh, those are, yeah, this is just visualization. So we run the experiment in lines. So we have line 1, line 2, line 3, line 4. So when now when you're visualizing your data, the software will just fill that entire height of the line. for each pixel. So you detected the signal in the center of that. So you have a spacing between the lines of, say, 10 microns or 20 microns, right? And so the just visualization software will fill in that gap. So you don't have data acquired between, there is a gap between the lines, right? So you acquire data, one line, then another line, another line. There is a gap between the lines that you didn't sample across, right? But software will fill in that information for you. So it will take the signal from that position in that line and spread it over the entire width of the line. So that's why it looks like that. When you have a lot of lines, it doesn't look, if you consider that pixelated. That's what the pixelated image looks like for us, because our pixels are rectangles and not squares. So you can imagine a pixelated, image with square pixels. This is a pixelated image with rectangular pixels.
Awesome. Any other questions? Okay, let's give a round of applause for Dr.
you.
Laskin. Thank you.